All right, good afternoon. I'm going to show you how to do one more protein sequence problem. A couple slides before we get that, just because I had the time and I want to show you why this stuff is important. We already talked about some of that. But the different chapters are actually involved in different kinds of stain removal. Everyone wants to get their laundry clean, and there's actually a lot of chemistry, even chemistry jobs, in making detergents. And we talked about SDS Page as a detergent, and so it's important to remember the primary purpose of detergents is to be able to have their hydrophobic tail mix with hydrophobic things, and then their hydrophilic head group mixes with the water, and that's able to pull the hydrophobic thing into the water to be washed away. It's all chapter 2. It really comes down to that. So we want to look at these different kinds of stains and the different ways of removing stains. If you get a greasy stain on your shirt, you want to use a stain stick, something hydrophobic, that will be a surfactant or detergent that will be able to dissolve it. For example, if you zoom in on these, so there's 3 different kinds of reactions here. The one in the middle is just using bleach, where you use oxygen, reactive oxygen species, to just break bonds like crazy. And so that's what the oxidizable stains are. Tea, coffee, and red wine, you want to bleach that. It will also take away the colored dyes in your clothes, so sometimes you want something a little more specific than that. And bleach won't do much for greasy stains. If you get butter on your shirt, you need to use a stain stick which has a detergent and surfactant in it. It might even have SDS in it as a way to be able to take those hydrophobic things and dissolve them in hydrophilic water. Now chapter 6 is going to talk about another thing. If you have blood, grass, or chocolate, those are protein-based stains. So to break those apart, you want to use a protease. Remember that proteases will break the amide bonds. And so you actually have special proteases in detergents that will take care of blood, grass, and chocolate stains. You don't want to use a stain stick necessarily. It might have enzymes in it, so don't quote me on this. But a stain stick that's good for greasy stains won't do much for enzymatic stains because they are fundamentally chemically different. The whole point of this class is for you to know what those differences are. And even concentration calculations, they might seem boring, but they're actually really important to understanding what's going on at the atomic level. Here's a quote from Bill Bryson. "If you could visit a cell, you wouldn't like it. Blown up to a scale at which atoms were about the size of peas, so an atom about this big, the cell itself would be a sphere, roughly half a mile across, like the size of the SPU campus. And it'd be supported by a framework of girders called the cytoskeleton. Millions of objects, and some will be as big as basketballs and others the size of cars, are basically moving around in 3 dimensions, moving as fast as bullets on that scale. There wouldn't be a place where you could stand without having all these things running into you thousands of times a second. So even for full-time occupancy inside of the cell is a hazardous place. A strand of DNA is on average attacked or damaged once every 8.4 seconds, 10,000 times in one day by chemicals and other reagents that just happen to run into it. That's why we have DNA repair enzymes, and that's going to be a topic for much later in the class. So to be able to think on this scale, think on the atomic scale, you can calculate what the concentration of a particular protein is inside a cell. And that's even homework problems that you've done. Typically, a cell will contain 20,000 different types of protein. Of these, about 2,000 types will be represented by at least 50,000 molecules. That means that even if we only count the molecules that we have more than 50,000 of them each, the total is a minimum of 50,000 times 2,000, which is 100 million protein molecules in each cell. That gives you an idea of the swarming immensity of biochemical activity within us. We're putting numbers on that, we're getting structures on that, we're telling something about where the atoms actually are. And even just a concentration calculation, an amino acid concentration calculation is what you need to do if you have phenylketonuria, which is actually a topic for later in the class. We'll talk about exactly what that is, but you see the phenyl in it. You don't have an enzyme that will break down your phenylalanine. So you need to eat foods that have less phenylalanine in it. There's a certain amount of phenylalanine you can handle. If you go over that, then you have too much, and your body gets overloaded. So what do? You weigh out your food, you calculate out what percentage of phenylalanine is in this, and you basically don't exceed a certain level. And so they have calculators and apps that help people with this disease to be able to calculate how much of this food can I eat, because you can calculate what percent of that protein is phenylalanine. And this is actually a life-saving app. But on the inside, it's just a regular old general chemistry calculation done with biochemical molecules. It's a test 1 kind of subject. So, with all that, I want to give you one more peptide sequence. This is one where you determine the peptide sequence not using Edmond degradation, but using reasoning. It's not 100% realistic, but that doesn't matter. The reasoning is 100% realistic. Okay? So, let's say that you get a test problem, and you say complete acid hydrolysis yields these amino acids. This is the first step where we say what are the pieces of the puzzle we have. And so what I do for this, and I don't know if my pointer, it'd be nice if I could type on this. But I'm just going to say, so what I'm going to do is I'm going to write down each of these. It's doing a weird thing with my pointer, but I'm going to try to put brackets around this, and I'm going to keep the plus there to remind myself I know that these are separately somewhere in the peptide, but I do not know their order yet. And so that's what I'm going to write down. I'm going to write down this with the pluses, and then when I know what their order is, I'm going to remove the pluses and have them in the actual order. So we move on to step 2. We treat them with FDNB, followed by the same complete acid hydrolysis that will tell us dinitrophenylalanine, DNP-ALA. Now, realize there's a little thing here. What is the N-terminal residue? And the N-terminal residue has had a dinitrophenyl group added to it. If it was phenylalanine, this would be dinitrophenylphenylalanine, but there's just one phenyl. So that means that it's dinitrophenylalanine. I can draw the little line right there. That means I know that my first molecule is A in my sequence, my N-terminal sequence, and I don't know anything else. So I'm going to keep those as pluses, and I'll write that down. Partial credit for just figuring this out. Then we're going to start adding our proteases. Here's a protease called carboxypeptidase, and it did not release an amino acid. Okay, that's weird. Carboxypeptidase releases the C-terminal amino acid in a linear peptide unless the last residue is pro. So I've got a proline among my pieces, so I'm going to consider it a high possibility that I have this. A goes to P. There's also a minor possibility that it could be circular. It's weird, but it can happen. You can condense, you can react the amino on one end with the acid on the other, and you can make it a loop. Some bacteria do that, and I think this showed up in one of your homeworks. But all this is to say, we don't know. So I'm just going to leave it as a question mark, as an unlikely question mark. Probably the last residue is pro, and we have a linear peptide. So now we get another piece of information. Cyanogen bromide actually cleaves it, gives 2 peptides, and one of them has serine, tryptophan, and proline. The other contains all the other ones. And cyanogen bromide, remember I will always tell you this, it cleaves on the carboxy side of methionine. So that means that this does not include methionine in it. Methionine is previous to this. You see the methionine is up here. It's the other amino acids. That means we actually can know 2 things. We can know that S and WP are together in the sequence. I'm going to put those in parentheses. And we know that the methionine must be in the middle, not at the end of the peptide. And so the SWP must be at the end of the peptide. Then I look at this and I say, oh, that includes the proline. And I'm already pretty suspicious that the proline is at the C terminus, And so this actually fits with that information that the CR, trip, and pro are the three residues at the C-terminus, and pro is probably the last. So this is what I know. I don't know the order of S and W yet, but we really are starting to narrow things down. So now treatment with chymotrypsin is going to do 3 peptides. We have the 3 peptides listed here, and chymotrypsin cleaves on the carboxyl side of things with aromatic rings, phenylalanine, tyrosine, and tryptophan. Well, I look at this, I have 2 cuts, and I'm going to look for my whole list and things with rings. What has a ring? I have 2 things with rings, and I have 3 peptides. So it sure looks like, yeah, none of these is at the C-terminus, probably. And so I have a cut next to my F and a cut next to my W. And lo and behold, I already know that my W is at my C-terminus, right? So I can actually go, I get this information from part E. I put this information together with that information, the previous slide, and I know that WSP, because I get a serine and proline together, those must be together, and the W must be over here, and the serine must be over there. In other words, I know now that it's MWSP, and the carboxy, this will cut it right here. The chymotrypsin will do this cut, whereas the cyanogen bromide did this cut. And so if I put my other information together, the FKSRA go together. And so if I have the M and W go together and the FKSRA go together, and I know my A is on my N-terminus, look at what we know now. We know we have an F and it's cutting here, a W and it's cutting there. The F must be at the C-terminal end of each of these individual peptides. And so we look back up, and the only thing we don't know is what's the order of the K, S, and R. Everything else we figured out. One more piece of information. We have 3 more peptides after treatment with trypsin, and trypsin cleaves on the carboxy side of lysine and arginine. Where are those? There's an arginine in this peptide, there's a lysine in that peptide, and there's nothing in this peptide, but we already know this is the C-terminus. What's the other piece of information we can get? I already know my AL is my N-terminus. So this allows me to just go through. I know I have these 3 peptides. I put that in the context of the previous one. I bring this over. I know that I have this brought over from the last step. And so it must be AR must be together. So I move the R over here. And then I have SK. I must be cutting after the K. So you see here, this gives evidence that I'm cutting here and I'm cutting there. That will give me these exact results. Those cuts give me those results. So then I finished it. I've determined the peptide sequence, and it's ARSK FMWSP. And all of this is, if the proline was actually part of a circular peptide, we would have gotten other peptides joined together previously. So now I can go back and say, I had a little question about that, but no, the weird thing is not happening. This is a normal sequence and linear peptide. And that's the test answer. 10 points out of 10. And that will be a good thing. If I give you something like that, that's how you reason through it. Okay, learn how to do these problems. You have several homework problems that are like this. I can guarantee you something's going to be on the test, probably at the end, because this is kind of complex and different, and I want to put those at the end of the test so that you can see that maybe you should focus on the straightforward stuff first, work your way up to this, or maybe you want to take on these first. It's up to you. All right? So good luck with this. I'm going to discard. And I'm going to post this on YouTube for y'all.